Hello, my name is Luis Barrera. I'm a senior at John Jay College, and my research investigates mitomycin C regulation of microRNA 5035P and MCF7 breast cancer cells. My mentor is Lissette Delgado-Cruzata, and I receive assistance from Dr. Yoel Rodriguez. We conduct work on breast cancer because, according to the World Health Organization, between 2015 and 2020, 7.8 million women alive were diagnosed with breast cancer, making it the world's most prevalent cancer for women. Mitomycin C is a natural chemotherapeutic drug isolated from the gram-negative bacteria Streptomyces caspitosis, used to treat different types of cancers, including breast. Studies have shown that mitomycin C binds to GC-rich regions of DNA and RNA. Among RNAs, microRNAs are a class of non-coding RNAs that serve as regulators of gene expression post-transcriptionally. Thus, deregulation of microRNA expression plays an important role in breast cancer tumors. One microRNA, microRNA 5035P, directly targets ZNF217, an oncogene that is frequently amplified in cancer, and it has shown that overexpression of microRNA 5035P suppresses MCF7 cell proliferation by directly targeting IGF-1R, an insulin growth receptor. While mitomycin C has been reported to potentially modify RNA molecules, it is not known if it can impact the levels of microRNAs such as 5035P. Here we investigate the effects of mitomycin C on microRNA 5035P expression levels in MCF7 breast cancer cells and explore potential mechanisms. We conduct in vitro experiments in which MCF7 breast cancer cells were treated with 50 micromoles of mitomycin C for 24 hours. After exposure, total RNA was extracted from these cells and control cells. We then determined the levels of microRNA 503-5P using reverse transcript and qPCR. We also predicted secondary and tertiary structures of microRNA 503-5P after retrieving the sequence from mRNA base. We did this by using RNA structure, RNA fold, simRNA web, and iFold RNA programs. The structure was chosen based on the most accurate nucleotide location, arrangement, and bonding with neighboring nucleotides. The 3D structures were generated using VMD software program. The structure with the most completed phosphate backbone was chosen for 3D representations. We found in Figure 5 the microRNA 503% relative expressions changed after the 24-hour exposure to the 50 micromoles of mitomycin C. We found that expression was significantly reduced after exposure when compared to the control. In Figure 6, we show the secondary structure of microRNA 503. The color of the bases indicates the base probability, as indicated in the legend. For example, the red represents 99% probability of the location and binding. In Figure 7, we show the microRNA 503 3D structures in three forms: a CPK representation in A, B, the licorice representation, and C, the molecular surface representation with cavities highlighted in yellow. These cavities represent the potential binding sites for chemical compounds. Throughout our work, we conclude that mitomycin C has a capability to impact microRNA function in the cell. In future work, we will carry simulations of the modified microRNA 503-5P molecule with the mitomycin C adducts to understand the mechanisms that mediate the disruption of microRNA function by mitomycin C. Thank you.